Welcome everyone to today's Explainer Masterclass. I am absolutely thrilled to have you here because today we are shrinking ourselves way down to explore one of the most fascinating environments in the known universe, the eukaryotic cell. We're going to take a grand tour of the microscopic factories that keep you, me, and literally every complex organism on Earth alive, breathing, and functioning. So grab your notebooks, get focused, and let's dive right into this.
Here's a quick look at our syllabus for today's masterclass. We'll start with studying the fundamental unit, move into the Eukaryotic Information Center, tour the endomembrane factory, check out the cellular energy transformers, and finally, look at the cell in action.
All right, Module 1, studying the fundamental unit. Before we can really get into the anatomy, we have to ask, how do researchers actually manage to isolate these structures that are completely invisible to the naked eye?
Well, the answer is this incredibly elegant technique called cell fractionation. It's basically a giant biological spin cycle. Biologists take a tube of disrupted cells and spin them in a centrifuge at increasing speeds. By playing with the g-forces and the time, they can systematically peel back layers of the cell based on size and density. Think about this timeline. After just 10 minutes at 1,000 times the force of gravity, the biggest pieces, the nuclei, settle out. Spin it for 20 minutes at 20,000 Gs, and boom, you've isolated the mitochondria. Push it all the way to 3 hours at a staggering 150,000 Gs, and you finally force those tiny, lightweight ribosomes down. It's a brilliant process of elimination.
Let's do a little data analysis here on cell size, because you might be wondering, why are cells microscopic in the first place? Why aren't we just made of one giant cell? Look at the math. A basic one-unit cell has a surface-to-volume ratio of 6. That ratio is absolutely crucial because the surface area, the membrane, is the only way nutrients get in and waste gets out. But if we scale that cell up to 5 units, the total volume skyrockets to 125, while the surface area only hits 150. That plummets our vital surface-to-volume ratio down to a mere 1.2. The data clearly shows that a cell that large would just suffocate and starve. They literally have to remain microscopic to survive.
Let's move to Module 2, the Eukaryotic Information Center. We are zooming right into the heart of the cell now. First off, let's pin down a foundational definition. The word eukaryotic comes from the Greek eu meaning true and karyon meaning kernel, or in this context, nucleus. So a eukaryotic cell literally means a cell that possesses a true nucleus. This is the exact opposite of prokaryotic bacteria, which evolved before the nucleus and just have their DNA floating around freely. That true nucleus, it is the hallmark of complex life. Inside that true nucleus is the master blueprint. If you stretched out the DNA in a typical human somatic cell, it'd be about 2 meters long. Yet it is coiled up so perfectly and efficiently that it forms exactly 46 discrete chromosomes. 46 volumes of your personal genetic encyclopedia.
Now, biology is full of surprises, right? So I want to call out a major biological exception here. While standard human cells contain 46 chromosomes, our reproductive cells, the eggs and sperm, are the explicit exception to the rule. They carry exactly half that number, 23 chromosomes. This is so that when they fuse during fertilization, the resulting human being hits that magic number of 46 again.
Now that we understand the blueprint, let's step out onto the factory floor. Module 3, the endomembrane factory.
There is a very clear division of labor right outside the nucleus in what we call the endoplasmic reticulum, or ER. We've got 2 distinct regions. The smooth ER completely lacks ribosomes, so it looks, well, smooth. Its job is synthesizing lipids and detoxifying poisons. Right next door is the rough ER, which is studded with thousands of protein-making ribosomes, giving it a really bumpy texture. The rough ER is your classic protein assembly line. It builds membranes and secretes proteins wrapped up in transport vesicles. Form, perfectly matching function.
Let's look at a concrete example of this factory network actually doing its thing. Take the process of phagocytosis, which is basically cellular eating. Step 1. The cell creates a lysosome, which is a specialized sac packed with highly active hydrolytic enzymes. Think of it as the cell's stomach. Step 2. The cell engulfs an outside particle into a food vacuole, and that lysosome docks and fuses right with it. Step 3. Those powerful enzymes deploy to digest the food particles into simple nutrients. It's an incredibly coordinated feeding and recycling mechanism.
But what happens when this precise machinery breaks down? We have to look at a rather tragic medical exception here. In a rare disorder called Tay-Sachs disease, just one single, crucial lipid-digesting enzyme in the lysosome is missing or inactive. Because that one specific microscopic worker is gone, lipids build up and create these devastating cellular traffic jams, specifically in the brain. It really highlights how absolutely critical every single piece of this system is to our survival.
Okay, running a sprawling factory with all that manufacturing and recycling requires massive power generation. Module 4, Cellular Energy Transformers.
Before we dive into the power plants themselves, we need to talk about the data surrounding where they actually came from. This is the endosymbiont theory, which suggests these energy transformers were once ancient independent bacteria that got swallowed by an evolutionary ancestor, and the data is super compelling. First, unlike other organelles, they have a double membrane, one from the original bacteria and one from the host that ate it. Second, they have their very own ribosomes and circular DNA, totally separate from the nucleus. And third, they literally grow and reproduce all on their own inside your cells. They're essentially a life form living inside a life form. It's wild.
So the two main power plants we're talking about are mitochondria and chloroplasts. Mitochondria are the engines of cellular respiration. They take in oxygen and generate ATP energy from the food we eat. Chloroplasts are the solar panels of the plant world, using photosynthesis to make sugar from sunlight. Now, while plant cells get to use both of these, animal cells rely completely on the mitochondria to keep the lights on.
We've made it to the final synthesis of our masterclass, Module 5, the cell in action. Let's see how all of this actually supports motion.
Let's look at cilia, those tiny microscopic hairs on the outside of cells. Motile cilia, the ones that swim or sweep mucus out of your lungs, are built on a very standard 9+2 pattern of microtubules. But check out this brilliant structural exception. We also have primary cilia. These feature a 9+0 pattern. Because they're missing that central pair of microtubules, they literally can't swim. So what do? They act as a rigid signal-receiving antenna, transmitting environmental data straight into the cell. One tiny structural tweak, and you completely change a motor into a sensor. As we wrap up today, I want you to really let this quote sink in. The cell is a living unit greater than the sum of its parts. We spent this whole explainer pulling the cell apart, right? Looking at the nucleus, the ER, the mitochondria, but none of these exist in isolation. Life arises from their integration.
So, I'm going to leave you with this final thought to chew on. Right now, as you listen to this, inside your body, a white blood cell called a macrophage is hunting down invading bacteria. Think about what has to happen. The nucleus has to send out RNA blueprints. The ribosomes have to translate them into proteins. The mitochondria must burn fuel to provide energy. The cytoskeleton physically drags the cell forward to grab the prey, and the lysosomes deploy for the final kill. All of this is happening blindly, simultaneously, perfectly, millions of times a day inside you. It's an absolute marvel of biology. Thank you so much for joining me for this masterclass. Keep asking those great questions, and I'll catch you in the next one.